Hello, everyone, and welcome. I'm James White, Collaborative Communications Director at CDD, and I'd like to thank you for joining us for our Q3 2025 product update webinar. It's Collaborative Drug Discovery. So before we get into today's updates, a few quick housekeeping notes. Please submit your product-specific questions in the Q&A tab, and we're going to address as many of those as we can in the dedicated Q&A at the end. For general comments or discussion, please feel free to use the tab chat, and hopefully we'll get a nice discussion going through the webinar. And before the Q&A, I want to mention that we're going to have a poll go up so you can request a one-on-one demo or more information. So high level 10,000 view of today, we're going to be covering some biologic and bioconjugate registration and visualization tools that we're rolling out, as well as a chemistry ELN workflow set of enhancements. So these updates are continuing to build on user feedback and designed to simplify the registration, experimental workflow processes, and make your research data more accessible and actionable. So let's kick it off with the latest developments in the biologics and bioconjugates. So these are playing a much larger role in modern research, and the industry is expanding. So we've brought those tools into CDD Vault so that you'll be able to take advantage of those. So we've added intuitive registration for complex biologics, including antibodies, peptides, as well as bioconjugates, flexible representations of the linkers and payloads and modifications on those bioconjugates, as well as clear visualization of the constructs, enabling you to review and share those structures at a glance for streamlined collaboration. We've also unified the data capture across both biologics and small molecules so your team can work in a single consistent environment. The goal here is to reduce the friction that researchers might face with large or hybrid molecules, and these are going to bring better structure, traceability, and visualizations to support both biologics and small molecule discovery. So really excited to intro Matthew, who's going to be doing our demo on these biologics. So please take it away, Matthew.
Hello, everyone, and welcome to our Q3 quarterly webinar update. My name is Matthew Harris, and I'm a customer engagement scientist here with CDD Vault. Today, I'll be reviewing some of our exciting updates to our macromolecule editor and some of our new registration capabilities surrounding cyclic peptides, antibodies, and antibody-drug conjugates. Now, going into our create a new and molecule option here, we'll focus on macromolecule editor first. Going into the launch structure editor now pulls up the editor that you would normally see for Catcher when you would draw your small molecules. Now from this view, we can click on this molecule option in the dropdown and select macromolecules. Now from this view, we have now the ability to start adding in our different sequences. As you see here in the top, you can add RNA, DNA, as well as peptides or say amino acid bases to the canvas here. So you can start building out your sequence right now from the canvas. However, if you do already have a sequence you would like to add to the canvas yourself, you can do so by simply clicking on the envelope here in the top left-hand corner, and then clicking on the paste from clipboard option. Now we do have a sequence that we can kind of play around with here. So if I just copy and paste it over now into the canvas, just a short RNA sequence that we'll be working with here. Now, once that gets pasted in, bottom left, you can see that there are different formats for which we do accept when you do add to the canvas. You can use a catcher file, Molfile V3000, sequence, which is what we have, FASTA, IDT, or HELM. And any one of those can be used for showing here in the canvas or for registration purposes as well. But we'll choose sequence for now and add to our canvas. Once that gets added, you'll be able to see that now we have our sequence of that RNA here on the canvas. Now, you can also, from this mode, which we call sequence mode, add other different sequence types. So again, we have an RNA sequence here, but let's say maybe we want to start adding some amino acids. We can click there on the peptide option, and then just start adding some in by typing them into the sequence. Once they're added here, you can see that now we have an almost hybrid molecule, which now contains an RNA sequence as well as amino acids. So from this view of the sequence mode, we'll call it, in the canvas, you can combine different sequence types to then start building out a unique macromolecule this way. Now, if we just remove these amino acid sequences, amino acid bases that we have here in our sequence, what we can also show is that if I select the highlight button again and highlight just a portion of my sequence here, I can now create an antisense strand just with what I've selected there as the kind of short sequence to create an anti-strand with. I could have also done that if I do the back button, if I had selected the entire sequence and created an antisense strand from that as well. So it's very easy to start adding these into your growing sequence if you want to add that here. Now, what you can also do from this sequence mode, there are other ways to visualize your macromolecule as you have it in the canvas. So outside of the sequence mode, there's also something that we call snake mode. Now, this now allows you to see the individual bases that were there, so AU, CG, but now we actually can look at the individual components of each of those bases. So we have the base, we have the sugar, as well as the phosphate. And those are all being shown here in these kind of different images. So we have a diamond, a square, and a circle representing those three different components. What you'll also see is if I hover over one of these, let's just say this sugar, you'll be able to see that there are the chemical structure is going to be shown for each of these different representations. So they're all here. So we have the sugar, the phosphate, and the base. So if you hover over one of these components, you'll be able to see the chemical structure that is below the picture representation here in the canvas. What's something else you can see here is that you'll notice that there are other kind of attachment points on this sugar in particular. You'll have an R1, R2, and R3 here that we're looking at, and those denote the different attachment points that you can have off of this one component. So one thing we like to say here with the macromolecules is that the way that they're being registered is that they're being registered in a chemically aware fashion. And what I mean when I say that is, as you can see, the correct bond formations and stereochemistry will be maintained within these macromolecules. You'll also be able to add and use any of these modified or unnatural bases in your sequences, as well as any, say, hybrid sequences, as we saw in the beginning where we added those amino acids to the RNA strand. So with the chemically aware registration, it allows for all of those different modalities and kind of information to be captured here within these macromolecules. Now, with these being chemically aware as well, you also have the ability to, if I just move this over here, you can also look at the chemical properties that you also have for these sequences. So if you click on the calculate properties here, you'll also be able to see that below, we do give you the chemical formula. We have the mass. We also have the composition of the sequence that's on the canvas here, as well as looking at the melting point and a few other things. So there are some chemical property informations that is captured within the sequences on the canvas here. And again, because they are chemically aware, we are able to calculate these things. Now, another thing that you can also see from these sequences is that if we go back into the sequence mode, and now, again, we'll choose the selector, we'll remove these, and now if we actually paste in a peptide sequence into our canvas here instead of that RNA sequence, we'll also be able to get the same thing with that calculate properties option. So as you see here, we have the chemical formula. We also have the mass, the isoelectric point, the extinction coefficient, hydrophobicity, as well as its composition. So regardless of what macromolecule you'll have here, you'll be able to have some of these basic calculations done for you based on what you have in your canvas. Now, another really nice thing about the macromolecule editor canvas here is that if I switch back over to snake mode, is that over to the right-hand side, you'll see that we have various other kind of libraries of modified and natural bases, whether they be amino acid. We have nucleotides. There's an option here called RNA Builder, which allows you to make a custom base by utilizing different sugars, bases, and phosphates to create a custom nucleic acid, as well as a chemical tab, which allows you to add chemical modifications. So think of, say, pegs of different varying lengths, or if you have a fluorophore, like say Cy5, those can very easily be added to the canvas. Now, let's say you wanted to add to your kind of peptide that you have here, some few bases that you have in our library. Let's say we choose this beta alanine. You can very easily click on one of these, add it to your canvas, And now they're kind of added to the canvas now, but not attached to our sequence yet. To attach them, what you'll do is choose this single bond option here. You'll click on the base you want to attach and drag it over to what you are attaching it to. And once that's complete, now the base, the beta alanine in particular, is added to the end here at the alanine. One other thing you can also do is, in addition to adding it to, say, a terminal amino acid or a terminal nucleic acid, if you're adding to that, is you can take a base and also add it to any other internal base that you may want to kind of add to the growing chain. The only thing here is that there should be an open attachment point, as we see here on the lysine, for the other base, so in this case, the beta alanine, to attach to. And the other really nice thing, which again goes back to the idea of everything being chemically aware, is that you can select an R1 or R2, in this case on the beta alanine, to then attach to the R3 on the lysine. So the end user can have full control over which attachment point they use to grow their sequences here. So really nice kind of way to show all of these attachments. But now that we've gone over some of the updates here with our macromolecules, I'd like to go through some of the updates that we now have with our registration process with, in particular, the cyclic peptides to start out with first. Now, going over here to our import data tab, I have an Excel file with some cyclic peptide sequences here. So what you'll do is in the drop-down list, you'll select the compose macromolecules from columns option, and you'll update. Now, once you have this selected, you'll be given a list of options here, and you'll want to select the cyclic peptide sequence in particular. Now, from here, again, it'll take the sequences that you have in your Excel file and give you the cyclic peptide representation. It'll start with the first amino acid that you have in your sequence, and then create the ring going in a counterclockwise motion from there. Now, few things that you can customize here in the registration process. First is being the orientation of your cyclic peptide. Again, like I said, it'll use the first amino acid there and go counterclockwise starting at the north cardinal direction by default. However, you can also change it if you'd like it to be starting from the east, the south, or the west. And then in addition, for the orientation, you can choose clockwise or anti-clockwise for how the depiction is for your sequence. So a few kind of ways you can change how the sequence gets registered here in Vault. Now, before we talk about the antibodies and ADCs, we'll get to that in a second, but just also to highlight the kind of flexibility here that you have with Vault with these different registration types is that when you have these cyclic peptides registered, you can have your data displayed as normal like you would with any other entity in the Explored Data Registration table. So what we're looking at here is a list of cyclic peptides that we have registered. They have some EC50 MIC data that is here in the table. And so you can have your data shown here in the table like you would normally have. But the, again, added feature that you have with Vault and our visualization tool is that this can now be brought into our visualization tool, and you can use this again like you would with any other entity that would get registered. So you can have the different plot types, you can have your different coloring, different binning, again, allowing you to draw your connections across different assays with this new registration modality of cyclic peptides. So very easy to create these visualizations right here within CDD Vault. Now, getting to the antibody registration, now this again is done from our import data tab if you want to do it in bulk, and you would select again from the dropdown the option for compose macromolecules from columns. Now, once you have that selected, now you can start to build your antibodies like you would with your other registration types, much like we do with the cyclic peptides or small molecules as you register them. So as you see here from our Excel file, we have a column to call out our heavy chain. So you would highlight that column first, select the antibody ADC option, and then specifically choose heavy and do the same for the light chain column. Select antibody/ADC and light is by default. And once you have those two selected, now your antibody gets built here using the compose macromolecule editor. Few things to talk about here with this sequence registration. First is you can also have these sequences registered in Vault beforehand, and you can just reference the ID number that you have here in the system. The one caveat to using that method, as you see here, molecule name, is that the sequences should be registered using our legacy sequence registration entity type. So it was that single letter code if you have used it before. If they're registered in that fashion, then you can very easily reference them if they are registered here in Vault. If not, as you see, you can just put your sequence into an Excel file and we'll be able to read that for you. The other is if you have custom bases in your sequences, you can also provide us with an SD file, put it into this area here on the right-hand side, and once you have those modified bases in your sequence, we'll be able to properly register your antibody by reading that SD file for those modified bases. Now, the other thing you can also do within your sequences is that as you see here, there's the option to have like different heavy and light chain sequences instead of just having a one heavy and one light chain sequence. So this opens the door to being able to register bispecific antibodies. So if you have different sequences, say you have two heavy chain sequences or two light chain sequences, or maybe one heavy and two light, whatever combination you may have, you can register that here. Instead of using just the distinct heavy and light chain options, you would then use the H1L1, H2L2 option here. And as you see in the Excel, you would essentially have a separate column for those different sequences. And then instead of using, again, the heavy light, you would just call out for heavy chain 1 would be H1, light chain 1, L1, and so on. So it's very easy to register those other sequences. And one final thing to note here with the registration of your antibodies is that if I look at the antibody preview over here to the right-hand side and hover over the first one, you'll see that there is annotation on my antibody when it will be registered in Vault, which calls out the various variable and constant regions that I have for my heavy and light chain. And this information is annotated within the sequences themselves that I'm registering. Now, our support team can help show you how the annotation looks like and get you started there. However, the more information you provide us, the more information we'll be able to register and show with your antibodies as you see here. So we can show the various regions. You can also annotate in your sequences where you have your disulfide bonds. So your bridges within your hinge region can also be denoted by those annotations in your sequences. So the more information you provide, the more detailed your antibody depiction will be. That's not to say, though, that if you don't have that information, that it can't be registered here in Vault. As you see here, these sequences are registered without the annotation of the variable and constant regions, so we will still register the Y-shape antibody for you. The colors you're seeing also are all of the distinct bases within the sequence that's registered, so you don't have the depiction or of the different regions, as well as you don't have the proper locations for your disulfide bonds. However, we'll still be able to register your antibody just fine with the sequence information. Now, taking this a step further, we also have the ability to register your ADCs or antibody drug conjugates. And so that's very easily done, again, from an Excel file where you would choose the composed macromolecules from columns and you would update. And the only extra step here is that when you go to map your heavy and light chain in particular, in this case, it's auto-recognize all of my columns, you'll also have one for your drug conjugate. And that's the distinction here where you see drug conjugate being chosen for this column that says drug. For the source of the structural information, you can provide it as a SMILES or Molfile. Molfile is what we have here. Or, much like with the sequences, if they're registered, you can have the linker payload registered in Vault already and just choose the option here if it is an ID number that will be recognized. There's also the option here to choose the different attachments. So this means that you can have up to 4 unique drugs associated on your antibody. Right now we just have one, but if you had multiple here in your Excel file, you would just choose the option. You can have different equivalents. As you'll notice, this first one has 8, and so that's what's being depicted in our antibody. The second one has 4, and that's what's being shown there. You can also choose the condensation option of your choosing here as well for how the drug conjugate will be attached to your antibody. So you have those options here. And again, looking at the antibody itself, you'll see that we do have the annotated regions of our antibody, and we also have our drug conjugates attached at the various locations. And again, much like the regions, the drug conjugates can also be annotated on the sequence so that they'll be attached at the proper locations. So it's very easy to start registering your antibodies and ADCs here in Vault, seeing as the interface is very similar to other registration types that we have here in the system. So, and with that, I can wrap up the biologics registration updates here with CDD Vault. We've looked at the new macromolecule editor, and the latest registration features for cyclic peptides, antibodies, and ADCs. This is all designed to make it easier to capture and work with these complex biomolecules here within CDD Vault. Thank you so much for joining, and if you have any questions, feel free to reach out, and we're always here to help you along the way. Thank you.
Fantastic. Thank you so much, Matthew. Next, we're going to be moving into the chemistry ELN workflow enhancements. So we're increasing efficiency and consistency here. You're going to have streamlined reaction setups with clearer input fields and automated property carry through, enhanced integrations between reactions and registered entities. So you reduce those duplications of effort that you might be experiencing. Better template management to support repeatable workflows and standardize your team's practices, as well as expanded visualization options to make it easier to review experimental context directly within your lab notebook. So these enhancements are going to help chemists capture data faster, cut down on manual steps, and maintain a cleaner, more connected record system within their projects. And delighted that Kelly is leading this demo on this chemistry ELN update. So please take it away, Kelly.
Yeah, thanks for that quick intro. I'm now going to share my screen and we'll take a look at some new ELN features that have been implemented recently. So what we're taking a look at here is an ELN entry with the list of some of these new ELN features. So we'll go through the publishing of templates, mentioning users, how to copy and paste chemical reactions, how you can then link your inventory samples directly to an ELN entry, how you can debit your inventory amount directly from an ELN page for a linked inventory sample, and then we'll take a look at some parallel synthesis examples. So I'm going to zoom out here and just show you the rest of this ELN entry that I have already pre-populated as the Vault administrator with some example information that I want my users to fill in using various different header sizes and colors to show the instructions for each of these sections. So once I have my entry set up the way I wanted to, I would then use this create template icon where I can click that and I can pick and choose which metadata fields I want to display for this particular template. So let's say I want to create a chemical reaction template here. I'm going to uncheck my assay type metadata field because I don't need that for a chemical reaction. I'm also going to make my NMR and my LCMS file a required field for my users so that they need to fill that in when they finalize these types of entries. So now I'm going to publish this template, and that template will now be available for my users to use when they create a brand new ELN page. I can do that either by going to the ELN index page or the Explore Data tab using this Create a New ELN Entry button. I will then be prompted to select a project, and then I can choose from my list of available templates which template I want to use for this particular ELN entry. I'm going to choose my chemical reaction. It creates the page and it populates all the information, including the title and the values that I had filled in for my metadata fields, along with the entire body of the entry. And now I can start making edits to my entry for my actual chemical synthesis. Now I'm going to switch over to a already pre-populated entry here where we've already gone through how to publish templates. Next, we'll take a look at user mentions. So you can mention the users in your vault on your ELN entry by using the @ symbol. And the workflow I'm going to show here is actually having a theoretical reaction set up that I want my supervisor to come in and take a look at. Maybe there is some safety information that they need to approve, maybe the scale of my reaction that they need to take a look at. So like I did here previously, what I'm going to do is use an @ symbol to call out my user, my supervisor, Jacqueline. And so Jacqueline will get an email with a link to this particular entry. Once she has gone through and maybe approved this reaction, she can fill in the approval date and time with a timestamp, that clock icon up the top, and maybe send a user mention back to me to let me know that this particular entry has been approved. So maybe that's one of the uses for your user mentions. You could also use them for collaborating on the same experiment with others, making some comments, and just calling out the other users who've been contributing to that entry. Now, this theoretical reaction workflow that I talked about, so I have my reaction set up, and now let's say it's been approved and I want to create another reaction with the actual measured amounts that I used when I measured out my reagents. So instead of having to recreate the whole entire stoichiometry table here, I'm going to use this copy reaction button in the top right-hand corner. I'm going to copy that reaction, scroll down to my measured reaction section, and use Control V on my keyboard to paste that reaction and stoichiometry table to the body of my entry. So now this new stoichiometry table is editable as well. I can go in and change the actual amounts that I measured. I hit enter and it fills in that amount. The other reagents will update accordingly, but I can also change these amounts. So maybe it was only 12.5 milligrams that I used. I can update those amounts. Now, I can associate the reagents that are already registered in my vault to their specific samples by clicking on their ID here. I need to specify a batch first, and then I can specify the sample that this reagent will be linked to. So now I've specified that sample. I can do the same for my other reagent here. I click on the pencil, I choose my batch, and I choose my sample. You can also link to samples in the notebook page using that @ symbol. So I can type in my ID here of my reagent. So PL12311-001-powder. And I can link to a sample this way as well. When I hover over the sample, it will show me the current amount that is available and the location of that particular sample. So actually, you'll notice that this batch number 1 only has 50 milligrams available. So I'm going to go in and change that amount in my stoichiometry table. So we'll put 50 milligrams in here, and that will adjust the rest of my sample values mass in the stoichiometry table. Once I link to a specific sample in my ELN page, I get this test tube icon that by clicking on will bring up a new inventory event. So I can debit a specific amount. The amount that gets pre-populated is the amount from the stoichiometry table that I used. Once I click update inventory, it would take that debit from my inventory amount. So I can do this individually for each of the reagents in my table, or I can do a bulk inventory event by clicking on this icon up in the top left-hand corner. I'm going to click on that. Anything that exceeds the current amount available, it will give me an alert for that. So let's go down to my last amount here. Oh, there's only 48 milligrams left. So I'm going to type 48 milligrams in here to use. We'll try that bulk debit again. And once I say yes, proceed, it will debit that amount from my inventory. And that test tube icon will then turn to this green color to show me how much of my inventory sample I currently have left. So you'll see that first sample, because I used all 48 milligrams, there are 0 milligrams left of that particular sample. All right, so just scrolling back up to the top, we looked at user mentions, we looked at copying and pasting reactions, linking inventory samples to our entry as well as to the stoichiometry table, and how to make debits from the inventory from that stoich table. So the last thing to take a look at are some parallel synthesis reactions. So I'm going to scroll down to the bottom here where I have a reaction set up. In order to make this a parallel reaction, I have to add an R group into my structure. So I clicked into the structure to open the structure editor. I'm going to go to my R1 icon over on the left, and I'm going to select the atom that I want to make my R1. Actually, I'm going to put R1 just as another reagent here. So I'm going to add R1 just to the canvas. I'm also going to add R1 to my product. And when both a reagent and a product have an R group, it's going to create a variable reagent and a variable product in my stoichiometry table. When I click into the entry, I can then implement my reagent product pairs manually. So I can click on this reagent, add reagent product pair. I can edit the structure and I can draw in my variable reagent or my variable product by editing the structure editor. I could also fill in the names of these reagents. The CAS number will sometimes pull up the reagent or the common name, or if it's a registered reagent in your vault, you could type in the ID of that reagent and you could implement that reagent or product directly into the stoichiometry table. So that's how to add them in manually. But let's say I want to add a bunch of different reagents at the same time. What I can do is upload a file to this stoichiometry table. So what I have here is an example of the Excel file that I would import to my stoich table, where I have a column for my reagent smile string and my product smile string. That's really all I need to input this into the stoichiometry table. I'm going to click upload and choose my file from my directory here. And let me just open up my file and edit that reagent. So I'm going to choose my file here from my directory. And it's going to automatically populate in my reagent as well as the product structure that I have from that Excel file that I just showed. So it'll go through each of these. You can then put in the mass of your products and it will calculate the yield of each of the products. If you want to register the reagent, the variable reagent, or the product, you also have these register links within the stoichiometry table to register each of those individually.
Thank you so much, Kelly. That wraps up our content. I hope you've enjoyed our walkthrough on the latest updated biologics, bioconjugates, and the chemistry ELN tools. Thank you so much for your attention and participation. Your input continues to drive our product roadmap, and we are really looking forward to your feedback so we continue that process. Before we open the Q&A, you should now see the option to complete the poll on your screen, so please take a moment if you'd like to request a personalized demo or any additional resources on the modules covered today or that you have questions about. So now we're shifting to our Q&A. We've already had some great questions come in, which we're about to talk about, but if you haven't seen yours yet or don't hear it, please submit it and we'll make sure that we get you those details, if not today, as quickly as possible. So let's begin with our first question.
All right. Yeah, hi everyone. Ralphie and Mitchell here on the Q&A side. Yeah, you want me to read the first question, Mitchell, or did you?
Yeah, I can maybe take the first one here, and we can get started. So yeah, thanks everyone for putting some questions into the chat here. It looks like the first one we have is coming back to the macromolecule editor from the first part of the presentation, and the question is, can the macromolecule editor work for other proteins? Thinking about glycoproteins here, with and without drugs loaded or attached. So great question, Ralphie, if you want to take that one.
Yeah, sure. Yeah, so the macromolecule editor is extremely flexible. We've designed it in a way that we don't really care what you put into it, as long as there are attachment points defined on any of your monomers, being proteins, amino acids, nucleic acids, or chemical compounds, whether they're larger strings of linkers or just a small molecule, a GalNAc warhead, whatever it is you're working on. So it's completely flexible. You can mix and match different types of monomers within a singular entity, and we'll be able to register all of that, because at the end of the day, the macromolecule editor translates whatever you do back into a V3000 mol file. So we have all of the chemical information under the hood that's getting registered in CDD Vault. All right, yeah, and I'll read the next one here off to Mitchell, kind of go back and forth. Yeah, next question also about the macromolecule editor. Can you add extinction coefficients for non-natural amino acids and use them in the calculation of the total protein 280 nanomolar extinction coefficient?
Yeah, it's another great question. So right now that calculation algorithm is built upon natural amino acids, but of course that's kind of the next logical place to go as we continue to develop that feature. So certainly a great question there.
All right.
All right. Moving down the list here, moving over to maybe some kind of integration workflow type questions here. We have, how well does CDD integrate with other systems?
That for me? All right.
Yeah, let's hear it, Ralphie.
Yeah, so I mean, kind of in the name, right? Collaborative drug discovery, we really try to enable scientists and end users to collaborate in any way necessary, right? So we realize that, we've been building a lot of great features in CDD Vault over the years. However, there's of course other systems within your total tech stack or ecosystem that you'd want to integrate with. So because of that, we have developed a RESTful API, which enables this integration with other systems, whether that's pushing and pulling data off of plate readers. We have some pre-existing pre-built integrations with systems like Spotfire. So yeah, you can look on our website. We have a whole partners page that'll show where we're actually integrating to, but the answer would be generally the API is your great friend there.
Sweet. All right, looks like we also have another question coming in about the macromolecule editor again, and this is really pertaining to modified nucleotides for different siRNA constructs, thinking about modified sugars, modified backbones, and sort of how that process is implemented into CDD Vault. Maybe I'll give a short answer, and Ralphie can add some more detail here. So the answer to that is yes, you can use all of your own custom sugars, bases, phosphates. You can register those types of entities in bulk or within the catcher window. There are also some kind of predefined libraries that may help you, build those types of things. So, Ralphie, if there's any other detail there.
Yeah, I mean, the other detail there is exactly as Mitchell said, but adding on to it, we also have plans, hopefully in the next quarter or so to address bulk composition of siRNAs. So similarly to what Matthew showed earlier with the antibody and ADC composition for multiple pieces, applying that to siRNAs is definitely on our roadmap as well, but you can do it today, on a one-by-one basis really well. All right, read the next one here. We have a few more on macromolecule editor, so yeah, I'll kind of
toss over to Mitchell one of them. I think they're kind of related here. So do you have, or can I as a CDD user admin define other condensed schematic visualization? So for the antibody and ADC, we currently have the Y shape, but the ask here is for other types of visualizations like fragments, SCFVs, Fab fragments, etc. So maybe I could
another great question. So right now it is kind of the default fragment there, or kind of template there. Obviously, this is a new feature we're building out, and as the features develop, we make them more customizable. So that kind of work is certainly on the roadmap. And Ralphie on the product team here can maybe touch on that a bit more.
Yeah, I mean, we've already been hearing a lot of great feedback from a lot of our excited customers, end users, developers. We have a few antibody ADC, experts in-house. So this is really the natural next step for us, not necessarily requiring those 4 strands, but building into other types of modalities. But of course, you can call out these regions on a full antibody today through your registration process.
And while we're on the macromolecule topic, maybe we'll go with the question here is, how flexible is the macromolecule editor to work with antibody fragments?
Antibody fragments. Yeah, so I think that's probably similar to the last request here, right? If you're looking at a specific portion of an antibody. So right now, the way we're building it is you're needing to take 4 chains. So whether they're, the same heavy, same light for monospecific, or you have a bispecific. We even have the ability to do like a trispecific or quad-specific antibody. It's more of a workaround and a place we plan to get to. But I think antibody fragments, portions of domains, that is an area we are leaning into. So what I would encourage you to do, if that's something that your team is interested in, please reach out to support. Help provide us with any types of examples so that we can help mark up these antibodies in the best way possible. Our real end goal here is to make something that's really usable to all of you. So yeah, any feedback on the process is greatly appreciated. All right, read the next one here. A couple more questions. We have some time. So yeah, if your question hasn't been answered yet, please do throw it in the chat. So kind of switching gears a little bit to, more chemistry oriented questions. We have a few here. So one is, do you have built-in experimental procedures for the chemistry experiments, like common named reactions?
Yeah, so in terms of kind of default layouts, right, we would probably recommend that if you are, running these different types of reactions, maybe repeatedly, that you might have a specific template to run a specific reaction type. Maybe you're running some Diels-Alder reaction every day. That's a super common workflow for your chemistry group. That might be a great use case for a template. Of course, you can look up all of your different reagents in the Stoich table and the catcher window. So probably a combination of building out the correct templates and kind of using those template and copy and paste features that we discussed earlier might be the best route there. And looks like we have one final question here. So again, if there are any lingering questions or more detail, feel free to ask. The final question is, can I create a chemistry ELN workflow where I loop in my colleagues?
Yeah, that's a great question. So really recently, we've released the ability to basically @mention your colleagues within an ELN entry. So while typing within the body of the ELN, just hit the @key and you'll see all of the colleagues on your team who have access to that project. You can click, call them out, they'll get an email notification to come into the entry. What we are really close to, so hopefully coming within the next quarter or so, is going to be comments within the ELN. So taking basically @mentioning one step further, similarly to what you have in Google Docs or a Word document, being able to highlight a section of an ELN entry, add a comment, call out a colleague within the comment, and there'll essentially be an entire comments panel on, the right-hand side of your ELN entry, really enabling that collaboration, right? So you can talk to one another, bounce ideas back and forth. It's kind of a separate object from the entry body, and that's really the collaborative space that we're envisioning there. So yeah, keep your eyes peeled for that.
All right. Well, it looks like we got through all of the questions there. Maybe we'll leave it open for a few more seconds here to see if anything else comes in. And Ralphie, maybe I'll pass it over to you for a closing remark here.
Awesome. Yeah, we really appreciate everybody's, attendance today. Thank you for your attention, your continued support, for all of the great questions. I think with that, we can wrap it up. Thank you guys for attending. Have a great rest of your day, and we look forward to seeing you next time. Bye all.